Welcome to the midterm review. These are all the topics for the year. The ones on the left show the topics that will be on the midterm. The ones over here on the right show what you'll do from the midterm to the end of the year. Those are the big topics. But for now, let's focus in on the midterm and let's look specifically at experimental design. So Jackie wants to know if people's pulse rates are going to increase if they eat food. She's going to test almonds to see if this is true. So we have to start with our prediction, our hypothesis. A hypothesis, like I accidentally said, is a prediction or a statement. It's never a question. If it has a question mark, it's not a hypothesis. Hypothesis must contain the dependent and independent variable in the experiment. We'll go over those in a moment. The hypothesis must also be measurable. You shouldn't use words like better. You should be very specific in what you should measure. And never have explanations in your hypothesis. No words like because. Those words are saved for the conclusion at the end of the study. So an example of a hypothesis, if people eat almonds, then their pulse will increase. We could change that and say their pulse will decrease. That's fine. Or we could say the pulse won't change at all. Nonetheless, it's a statement. It's our prediction as to what's going to happen. Next up is we have to identify the independent and dependent variables. The independent variable of an experiment is the thing that you're going to test. You're testing to see if almonds are going to cause a change in pulse. So almonds would be your independent variable. The dependent variable is going to be your data, your results, what you measure. You want to see if almonds have an effect. You want to see if they affect what? You want to see if almonds affect your pulse. So you're going to measure your pulse before you eat almonds and after. So your pulse rate will be your dependent variable, the thing that you're going to measure. And our hypothesis does have our independent and dependent variable. There's no question mark. So far, so good. We said it must be measurable. We could measure how many a person's pulse rate, how many beats per minute, how much their heart beats. What's an example of not measurable? I'm going to go a little outlandish. This experiment specifically is a little harder to do. Let's say if you eat almonds, that's better for your pulse. How can you measure better? Like eating almonds will make my pulse be better. That's ridiculous. You can't measure better. And there's no because, there's no explanations. That's for the conclusion part of a study. This hypothesis is perfect. Now we have to set up our experiment to test and see if our hypothesis is true. For that, we need two groups, a control group and experimental. The experimental appropriately receives the thing that you're experimenting with. So if you're experimenting with almonds, that's going to go in the experimental group. People in that group will eat almonds. The control group, just there for comparison, and you'll see what we mean later on. It does not get the thing you're testing. People in this group won't get almonds. This is the only difference. Literally, you could have one group of people, they could take their pulse before they eat almonds. That would be the control data. And then have them eat almonds, then take their pulse after they eat almonds. And that would be the experimental data. We're going to do two separate groups, though. Everything else has to be the same. The same type of people. If you have 100 people, they both have to be either boys or girls. They have to be the same. They have to be the same age. If you have a control group of 22 and experimental age 75, wow. that could throw off your results. That could be a variable that could influence your pulse. 7 hours of sleep in the control, 7 hours in the experimental. Let's say the experimental had 1 hour. That could throw off their pulse also. So we're trying to control as many variables as possible to make sure nothing else influences the pulse rate besides the almonds. So the more variables that you control, the more accurate the experiment and the data will be. So let's take a look at some results. Our dependent variable. The experimental group, people who ate those almonds, they took their pulse and it was 84 beats per minute. Did the almonds cause the pulse rate to increase? I don't know about you, but I can't tell. 84 beats per minute? We need a group to compare to, a group that did not eat almonds. Perfect, we got that. Our control group, our comparison group, doesn't get the almonds. They have 73. So our pulse, or the pulse rate, increased due to the almonds. And we could say it increased due to the almonds because we controlled a lot of variables. But that stinks. Our hypothesis is wrong. We said there'd be no change. That's not a bad thing. If your hypothesis is shown to be wrong, you still get valuable data. Now you could design a more accurate or more concise hypothesis, a more accurate experiment. Overall, though, you still learn information. You learn that almonds have an effect on pulse. Another quick little one. An experiment was carried out to answer the question, does the pH of water affect the growth of radish plants? So the independent variable, the thing you're testing to see if pH of water causes radish plants to change how they grow. The dependent variable is going to be what you measure, your results. So you're seeing if pH of water has an effect on the growth of plants. So the growth of the radish plants would be your dependent variable. Specifically, when we refer to growth, we're going to collect data. We're going to collect the heights of the radish plants. It says in that last sentence. So growth refers to height. That's the data we're going to collect. And I threw in centimeters there because you should always have units. And how could you make this more accurate, this experiment? Have more variables controlled. It says it in the little thing, there's a lot, same amount of water, same intensity of light. We're just going to say, do more, have more variables controlled. Run over a longer period of time. It says 2 weeks, make it 10 weeks. If you can't do 10 weeks, repeat the experiment. Set up 1 billion groups, not 1 billion, set up like 100 groups, 100 group of 10 plants. And use more test subjects. Instead of using 10 plants, use 100 plants. All those things will give you more data, which means you'll have a more accurate conclusion, or more reliable conclusion. So why is a longer-term study better or more accepted? Because you have more data. We just said that. This is just showing that in a different format. Longer-term study will give you more data, and if you have more information, you can make a more accurate conclusion. Like if somebody got a 50 on a test, that student can't do well. That was one test. They got a 50 on one test and got 95 pluses on the rest of the whole year's tests. That student's pretty darn good. You need more data, more accurate. And we said this, if the data doesn't support your hypothesis, you could still use that information to develop a more accurate experiment, a more accurate hypothesis. What exactly do we mean? Maybe you missed something, a variable that you didn't think would have an effect. So maybe control more variables, maybe have a new dependent variable, measure something different. And you could also develop, you said, a more accurate hypothesis. So experimental design we got down. Let's take a look at graphing. So when you do a graph, the independent variable is always in the bottom, dependent variable is always in the side. Think of a building in the ground. The building depends on the ground. The ground does not depend on the building. The ground moves, the building falls. So the building depends on the ground. The ground's independent, the building's dependent. Little trick to remember which axis is dependent and independent. Now we have to make our scales. We have to put all these years down there. You must have the lowest and the highest value. You don't have to have the exact numbers that are written in the data table on your scale. You just have to make sure they're on there somewhere. So if you include the lowest and the highest, you're going to have to include all the numbers in between. And you don't have to start at 0. If you start at 0, let's do a bad scale here. 0, 1880, 1890, and 1900. That's a disgusting scale. This first little box, or space, is worth 1,880 years. The second space is worth 1 year. Third space is worth 1 year. That's a terrible scale. Scales must increase and decrease by the same increments or same values. Just like a ruler. Each little space on a ruler, on a metric ruler, is a millimeter. And each little space on an English unit ruler, I believe, is 1/16 of an inch. They're all worth the same amount. Your scale should be worth the same for each space too. Never draw this little diagonal line outward and then put 0. You're saying both axes start with 0. So let's try and draw a scale here. Making each space worth 10, but it won't fit. I mean, I could start at 1870, but it doesn't fit 1990 on there. So I'm going to have to actually start at 1880, make each space worth 10, and it gets up to 1990. That will fit. Each space is worth 10 years. So it's evenly spaced. We're looking good. Just adding in what we just discussed. Each space must be worth the same increments up and down. Notice we don't have the year 1890 on this scale, but we know exactly where 1890 is on that scale. You don't have to write it. So our lowest and highest, 3 and 41, we have to include those numbers. You're probably going to start the scale at 0. You don't have to. If you start at 0, make each space worth 2. It's not going to fit. Let's try a different number. Let's make each space worth, 5. So every 2 spaces is worth 10. We'll fit all the numbers, and we're going to use just about half the graph. You want to try and use numbers like 1s, 2s, 5s, 10s. You don't want to go up by 3s or 6 or 7s. It's going to be very hard to find your data points. Once you get the scales done, you're going to plot all your data points. And once you got your data points plotted, you're going to use a ruler to make a straight line or the edge of a piece of paper. And remember, never draw an arrow at the end, because you're saying that you have data until forever. Nobody has data until forever. So never draw an arrow. The whole graph is wrong. You could only graph data that you have. So in some graphs, you'll see the line does not have to go to the origin, means 0, 0. Only graph the data you have. Just connect the dots. on to the microscopes. We're going to look at a compound light microscope. Microscopes are used in the labs, and they're used to view cells. It's called compound because you're going to look through two lenses. And in order to see your specimen, which is on the stage, you need the lamp, which is underneath. It shines light up. So when you have the light on, you're going to look through two lenses. The first one is going to be the eyepiece. That's what you put your eye on. The second is going to be next to the specimen or the object, called the objective lenses. And you have a low, high power objective. To get the total magnification of the object, you're going to multiply the eyepiece and the objective. If the eyepiece is 10 and the objective is 40, total magnification is 400. As you're viewing it, or once you put the specimen on the stage, you're going to need help finding the specimen. So you're going to use the coarse adjustment while you're under low power, because under low power, you're going to find the thing you're looking for. The coarse adjustment will move the stage up and down to help you find the object. Since it moves the stage up and down, you never use it under high power, because under high power, the objective lens is so close to the slide, if the stage moves up and down, it could break the lens and or the slide. The fine adjustment wheel, the small one, will help make the object more clear. It will increase the resolution of what you're looking at. And you can use this under low power and high power. It does not move the stage and won't damage the microscope. If the image is a little dark or too bright, you're going to use the diaphragm. It controls the amount of light that's let through, so it could increase or decrease the brightness. It could increase or decrease the contrast. So let's say you find your cell, it's off to the upper right. How would you move this slide in order to get that cell into the center? You're going to move the slide to the right and up. And when you're looking, it's going to look like the cell is moving to the left and down. Since there's mirrors and lenses used, things will appear to move backwards. Why center it in the first place? I found it. Well, when you zoom in, under low power, you have a real large field of view. You see a lot. Like if you have your hand far away from your face, but when you zoom in, you see less of your hand when you put your hand on your face. The high power field of view, the field of view is small. You see less because you're zooming in. So the yellow is what you'll see under high power. You're not going to see the cell. So if it's not centered and you zoom in, you're not going to see what you want to see. All right, we found the spot. So let's talk about some changes. What happens when you go from low to high power? Under low power, the objects or the specimens, they're super small. Under high power, the object or the specimen, huge. Look at the size difference of those cells. The diameter, meaning the field of view, is very large under low power, like it's 1.6 millimeters. Under high power, it's only about 0.4 millimeters, 4 times as small. So the field of view on a low power is very large. You see a lot more when you're zoomed out. Field of view under high is very small. Under low power, things seem very bright. Under high power, things are very dark, and you're going to use the diaphragm to brighten things up. Low power, things are clear. High power, things get blurry. You use the fine adjustment to make it clear. And under low power, you see a lot of cells and they're small. High power, you see less cells and they're big. So let's find out the field of view. When you look under a microscope, these are millimeter lines on a ruler, the smallest lines you could possibly have on a ruler. And from 1 millimeter line to the next, the space between, that's 1 millimeter. So from here to here is 1 millimeter, from here to here is another millimeter. This field of view is 2 millimeters. To convert that to micrometers, which you will have to do, set up this little staircase, 3 steps up, 3 steps down. This is microns down here, or micrometers. This is millimeters. To go from millimeters to micrometers, you have to go 1, 2, 3 steps to the right. You're going to move the decimal point 3 spaces to the right. Those little holes or rungs will get zeros. In 2 millimeters, there are 2,000 microns. The field of view for here, in millimeters and micrometers, we have 1, 2, 3 and a half. There's about 3.5 millimeters. And if each millimeter has 1,000 micrometers, just times millimeters by 1,000 to get 3,500 microns. Determining the size of a cell. Let's say you have a piece of bread, 10 feet long, big piece. You cut it in 2 pieces. How big is each half? Easy, 5 feet. That math that you just did in your head is the math you used to figure out the size of a cell. You took the total length, 10 feet, and divided it by the number of pieces. 10 divided by 2 gives you 5 feet per piece. Using the same math over here, the total length is 2,000 micrometers cut up into 5 pieces, or 5 cells. Each cell is 400 micrometers. Over here, total length, 4,000 microns, and you could fit about 5 cells in there. So 4,000 total field of view divided by the number of cells gives you about 800 microns for each cell, 0.8 millimeters. So we got graphing done, measurement, microscopes, all these life functions to the end. We're going to group these all together, and we're going to go pretty crazy with this. So buckle in. Cells, they're the smallest living thing that can carry out life functions, also known as metabolism. So all the life functions together, all your chemical reactions is called metabolism. Life functions is just simply the processes that make something living. They're GERNETs: growth, respiration, reproduction, regulation, nutrition, excretion, transport, and living things need to do synthesis. These are the 8 life functions all things must do. Cells, they're also the basic unit of structure and function, the basic structure of life. If you want to build a living tissue, you need cells. So a whole bunch of cells together make a tissue. A whole bunch of tissues together make organs, a group of similar tissues. Similar organs together make an organ system, a group of similar organs that carry out a similar purpose. And then all the organ systems together make a full organism. For an organism like you or me, our life functions are carried out by our organ systems, our cardiovascular system and whatnot. Cells, their life functions are carried out by things that are smaller than them. Things called organelles, like mitochondria or ribosomes, little things inside the cell. So we know everything needs nutrients to carry out life functions. You need nutrients to live. Proteins, called polypeptides, they're broken down or hydrolyzed, undergo hydrolysis, into amino acids. And amino acids diffuse into the cell, and they're going to go to the ribosome. The ribosome uses amino acids to make new proteins. Ribosomes are in charge of protein synthesis. Those amino acids are bonded together using peptide bonds to make a long chain of amino acids with many peptide bonds. Hence the name polypeptide, many peptide bonds. Let's talk about proteins. The order and the amount, specifically the order of amino acids, determines a protein's shape, and the shape of a protein determines its function. I can't tell you how important that is. Make sure you know that. So here's a little protein. Here are some things that could go into that protein. Letter A goes into the protein. We know it's not B. We know the function of that protein because the shape of the protein. The shape told us the function. Proteins, however, they could lose their shape. When proteins lose their shape, and therefore their function, that's called denaturation, meaning protein denatures. And there are certain conditions that can cause this to happen. So right now we know this protein will take in that little triangle or square. But if it's denatured, it can't. The shape is not there. It lost its shape, it lost its function. That protein's permanently denatured. So what causes denaturation of proteins? What causes a steak to go from red to brown? Heat, when it's too hot. When you have a high fever, it's very dangerous because you could denature the proteins in your body. Too acidic. If you get acid spilled on you, it can cause your skin to come off because it will denature the protein collagen that keeps your skin on your body and keeps your skin tight. Too basic. It's the main thing in cleaning supplies. Ammonia, which is super basic. It kills germs, viruses, and bacteria. What about too cold? Enzymes and proteins, they'll just work slower. They will not denature. Let's take a look at a few graphs here. This graph is going to show temperature and enzyme activity. Enzyme or protein activity. What's the range in which this enzyme works? About 25 to 70. It works from 25 to 7 degrees. What's the optimal temperature? You could always pause the video and try and answer it. Right there, that's where the most activity happens. It works best at about 47 degrees. About 47 degrees Celsius. A, B, and C, which one of those letters show a protein or this protein denaturing? A, it just works slower because that's too cold. B, definitely not. That's the optimal. That's the best temperature. C, too hot. That's when it denatures. All right, well, let's look at the other factor. Let's look at pH. I have a pH little graph here. It goes from pH of 3 to 11. pH scale does go from 0 or 0.1 all the way to 14. We're looking at a section of it. Enzyme A and B. Which enzyme works in a basic environment only? Or better? Basic is from 7 to 14. BBB, the bigger the number, the more basic the substance, and it turns litmus paper blue. The lower the number, the more acidic, and it turns litmus paper red. 7's neutral. Enzyme B only works in a basic environment. So different proteins, for example, like enzymes, work in different pH solutions and in different temperatures. All right, let's go back to where we started. Proteins, they're broken to amino acids. New proteins, examples are hormones, enzymes, antibodies, and receptors. Here, little acronym. Enzymes are also biological catalysts. Why? Because enzymes speed up hydrolysis reactions and dehydration synthesis reactions. Synthesis means to make or build. So enzymes speed up chemical reactions. If you're ever asked what enzymes do, that's why they're called catalysts. Hydrolysis is breakdown and synthesis is buildup, specifically dehydration synthesis. So this is a hydrolysis reaction. Hydrolysis, meaning water splits. You add water to break things down. That's why you drink tea or coffee after food, because it helps you break down material. The enzyme that breaks down protein is a category called protease. Examples of proteases are pepsin and trypsin. Pepsin works in an acidic environment. Trypsin works in an optimal pH of 7, around neutral. Pepsin works in the stomach. Trypsin works in the small intestine. All right, let's make this cell membrane a little longer. Let's look at carbs. Carbs, also known as polysaccharides. Poly meaning many, saccharides meaning sugars. Can you guess what carbohydrates are made of? Examples of carbs are starch. That's how plants store extra sugar. They store a whole bunch of sugar in a long chain called starch. Cellulose. This is the carbohydrate or polysaccharide that makes plant cell walls. Glycogen is how you and I store extra sugar, how animals store extra sugar. Carbohydrates or polysaccharides are broken down or hydrolyzed into simple sugars, also called glucose, also called monosaccharides, meaning one sugar. And sugars have a specific look to them. Here's a long chain of sugars, a bunch of little hexagons joined together by glycosidic bonds. The little hexagons, they're sugars. When they're broken down into single geometric rings, individual sugars. These individual little sugars, like glucose, C6H12O6, they're organic. So are carbohydrates. So are proteins, because they contain carbon and hydrogen. Carbon and hydrogen together means you're organic. And CH bonds, carbon-hydrogen bonds, store chemical energy. So simple sugars, they will diffuse into the cell, and they're going to go to this thing, this cell organelle, called the mitochondria. And the mitochondria is going to rip energy or extract energy out of glucose and make usable energy called ATP. ATP is usable energy, and it's used for metabolism. So here it is, ATP. But if you only use glucose, you're only making 2 ATP. You'll also make carbon dioxide. And if you just use glucose, you'll make lactic acid. It makes you feel sore, tired, or fatigued. In yeast, they'll make alcohol. 2 ATP is not enough. If you use oxygen, you'll make 36 ATP, 18 times more. And you won't make lactic acid or alcohol, you'll make the liquid water instead. This process of converting glucose into ATP is called cellular respiration. And I'm just going to draw out the equations for you so you can see all possible ways this is carried out. So first shows just glucose being used makes 2 ATP, carbon dioxide, lactic acid. Second, just glucose, make 2 ATP, carbon dioxide, and alcohol. These are both anaerobic, meaning without oxygen respiration, also known as fermentation, lactic acid fermentation or alcohol fermentation. Remember, lactic acid makes you feel fatigued. You can do respiration this way. Using oxygen, you'll make 36 ATP, carbon dioxide, and water. Aerobic, meaning oxygen respiration, is when you use oxygen to make ATP. And every time ATP is made, CO2 is made. So if you make ATP, which is every single living thing, you make CO2, because ATP and CO2 are always made together. All organisms make ATP to carry out life functions. So all organisms do cellular respiration. Therefore, everything, including plants, make CO2. But plants, they're going to use the CO2 they make to do this process, photosynthesis. And let's go on a tangent. Let's draw a little plant cell here. Here's our cell wall. Here's our cell membrane. And here is the organelle that carries out photosynthesis. You know it as the chloroplast. You need sunlight for photosynthesis, solar energy. You also need carbon dioxide, and you need water. Those things, if you give them to a plant, they're going to grow because plants will be able to make glucose, C6H12O6. They'll be able to make their own food. They'll also make oxygen as a byproduct. This whole process is photosynthesis, where solar energy is converted into stored chemical energy. And this is done, photosynthesis, by the organelle called the chloroplast. So the equation, sunlight plus CO2 plus water, makes glucose and oxygen. In other words, inorganic substances are turned into organic substances. Remember, organic, carbon plus hydrogen. Or solar energy is converted into stored chemical energy. Different ways to look at the process of photosynthesis. Now, plants, they make glucose not for fun, not for us. They make glucose so they could use it to make their own energy. So since they make glucose, their own food, they're called autotrophs, producers. And that glucose is made for a reason. It has stored chemical energy, and you can't use that. So it has to be converted by the mitochondria to make ATP. And whenever ATP is made, CO2 is made. But only 2 ATP are made if you use just glucose. So plants do use oxygen to make 36 ATP. And they still make carbon dioxide. But they're going to reuse the CO2. They're going to take the CO2 and recycle it or reuse it for more photosynthesis. All right, so again, that last waste product is water. ATP, we know, is usable energy used for metabolism. But this oxygen, the extra oxygen plants make, and extra water the plants make, how do they get rid of this stuff? They don't just hold on to oxygen because that'd be bad for us. We'd be dead. So how does the oxygen and water leave a plant? And how does CO2 get in the plant? For that, we have to look at a leaf. On the bottom of a leaf, the underside, and these little pores or holes. This is what they look like. This is where the oxygen and the water will eventually leave. Oxygen leaves, water leaves, and this is where CO2 will go in. This opening or hole is called the stomata or stoma. The cells on the side that guard it are called guard cells. Guard cells open and close the stomata. So this shows a wet day. The guard cells open the stoma on a wet day. On a dry day, there's less water in the cells and they become flaccid. They deflate. And the guard cells close the stoma to prevent water loss. So the guard cells, they kind of regulate or prevent water loss. And they also regulate the exchange of gases. All in order to maintain homeostasis or dynamic equilibrium. Where does gas exchange happen in humans? It happens in the stoma and leaves. What about us? For that, we have to look at a human lung. The human lung is like a sponge. It's filled with little air sacs called alveoli, or alveolus, singular. Here's the alveoli, and here's a blood vessel that's around the alveoli. Blood coming to the lung and to the alveoli is filled with carbon dioxide. The heart purposely pumps blood to the lungs because it has a lot of CO2. And you take a breath of oxygen in, oxygen will diffuse from high to low concentration into the blood. CO2 will diffuse from high to low concentration out of the blood. This blood vessel is one cell thick, just like the alveoli. The blood vessel is called a capillary. Being one cell thick allows for diffusion. All that oxygen will then go to the body for aerobic cellular respiration. All right, back to our cell membrane, just making it a little longer. We're going to look at fats, also called lipids. They're chemically digested into fatty acids and glycerol, the glycerol backbone. They're digested chemically by the enzyme lipase. Enzymes end in ase. Let's go back up real quick to the carbohydrates because we forgot to label that enzyme that chemically digests carbs. That's called amylase, after starch, called amylose. All right, back to our fats. Those fatty acids and glycerol, they're very rich in carbon and hydrogen. Fatty acid is just a long chain of carbon-hydrogen bonds. What does that tell you? There's a lot of carbon-hydrogen bonds. That means there's a ton of energy. So the more carbon-hydrogen bonds you have, the more energy you have. There's a lot more CH bonds, a lot more energy in fat than sugar. Fats are generally like a long-term energy storage. Carbs are more of a short-term, immediate energy storage or use. So let's see what happened to those fatty acids once they get into the cell. They're going to go to the organelle called the smooth ER, the smooth endoplasmic reticulum, and they're going to be synthesized into new fats, either storage fats in your adipose or fat cells, or make things like membrane fats. Those are called, or they look like this, and they're called phospholipids. Phosphate is a little circle head. The lipid tails are those little jellyfish feet that you see. And there's two layers of them. There's two layers of phospholipids. Cell membrane is called a phospholipid bilayer. It's also a semi-permeable membrane, which we will get to. Phospholipid bilayer, the cell membrane, also has other structures in it. These great big specific shaped structures. These shapes determine their function. These great big structures are called proteins. We have a carrier protein, a receptor, and a channel protein. So the cell membrane is made of phospholipids mostly, and also made of proteins. This is our receptor. This is our carrier protein, uses ATP to shove things in the cell. And this is our channel protein, a little opening to facilitate diffusion. So let's look at a cell. We got some oxygen outside, a little bit in. Some carbon dioxide around. We got some, let's reverse that. We got some starch in the cell, some glucose in the cell. And let's put some sodium out there. Sodium comes from the breakdown of salt. When salt hits water, it's broken down to sodium ions and chlorine ions. Oxygen is going to actually diffuse into the cell. Why? Because it wants to go from a high to a low concentration. So oxygen will move until there's 3 in and 3 out. There's no high, there's no low. Everything is equal. Equilibrium is established. And at equilibrium, things move equally. Carbon dioxide is going to move out from high to low concentration until it's equal. Something like starch wants to move out, but it can't because it's too big. Big things can't diffuse. Glucose, actually before that, how do you know if starch is present? You're going to use a chemical indicator called iodine. Iodine will tell you if starch is present. How? By changing color. It will actually change color from amber to a black, a purple black. Back to glucose. Glucose wants to get out of the cell. It wants to go from high to low. And it can because it's small enough. Glucose is a building block of starch. So it goes from high to low, of course, until it's equal. How can you tell if glucose is present, though? You're going to use another chemical indicator, Benedict's solution. How is it going to tell you if glucose is present? It's going to change color also, but you have to heat it. It's going to change from blue to an orange when heated. Sodium can't diffuse in because it's small enough, but it's charged. It's a charged molecule called an ion. So things that are charged can't diffuse. They're going to need actually special carrier proteins to push them in, which uses ATP. So let's take a look at this whole transport. There's two types, passive versus active. Passive transport's also known as diffusion. Small and uncharged material will move from areas of high concentration to areas of low concentration with no energy used at all. Active transport, small uncharged things will move from areas of low concentration to high concentration. That's going to need ATP. It will also move charged material through the membrane. And it will use the carrier proteins to do that. Active transport could also get large material into or out of the cell by processes called endo, meaning entering, or exocytosis. Let's take a quick glance at those. This shows a big molecule trying to get in. The whole membrane has to bend around it to get it inside, and that big piece of food is now inside a vacuole where it will be digested by a lysosome. This shows endocytosis. Going the opposite way shows exocytosis, if you do the pictures in reverse. So all these things need ATP, hence the name active transport, uses energy. And here's a little way to help you remember them. Let's say you have a tire. You have a high concentration of air in, a low concentration of air out. If there's a hole in the tire, air is going to move out. No problem. No energy needed. But if you want to get air back into the tire, you need ATP. You need energy. You have to go to a pump. So going from low to high, you need energy. High to low, you don't. If that doesn't work, think of a person on a bike going up a hill, really steep hill. This is up high and this is down low. The person goes up the hill from low to high, they're going to have to pedal for their life. They need ATP going from low to high. But if you go from high to low, you might have to use the brakes, but no energy is going to be used going down that hill. So passive transport or diffusion, there's a special name for the diffusion of water. It's called osmosis. And we have to take a little closer look at this. Let's say you have an animal cell, no cell wall, and there's 5% salt inside or sodium, 10% sodium out. Can't diffuse. The rest inside the cell, 95% is water. The rest of the 90% outside the cell is water. Both add up to 100. Sodium can't get in, but water can diffuse. Water will diffuse out of the cell from high to low. This is a saltwater solution or a hypertonic solution. And since water leaves the cell becomes dehydrated. meaning it's losing its water. The cell membrane shrinks, the cytoplasm is reduced, the cell is dehydrated. What happens in a plant cell? Let's put the plant cell in the same solution. You have the cell wall, the inner thing is called the cell membrane. So 10% salt out, 5% salt in, 95% water in, 90% water out. We're in a saltwater solution. Water is clearly going to diffuse out of the cell. What's going to happen to the plant cell? The cell membrane is going to shrink. Cell wall does not move. It's like a cardboard box and a balloon in the box. If you deflate the balloon shrinks, but the box stays the same. So cell membrane shrinks, cell wall remains the same, and the cytoplasm is reduced. Let me color in the cytoplasm to show you where the cytoplasm is. Cytoplasm will be red, because you did this in the diffusion through a membrane lab, looking at a red onion cell with red cytoplasm. So the cytoplasm you can clearly see is much less on the right after the cell was dehydrated. There is no cytoplasm between the cell wall and the cell membrane. So how could you get more of the dots out of the cell, going from low to high? You can use ATP. That's active transport. All right, getting towards the end here. Plant cells versus animal cells. Let's do some differences. Plant cells, they're autotrophic, meaning they make their own food, also called producers. The food they make is glucose. Glucose is made by a chloroplast by the process called photosynthesis. Only happens in autotrophs. Plants don't move, so they're going to be autotrophic. They have to make their own food. They have chloroplasts to do that. Since they don't move, they have a cell wall for extra protection and to give them structure. They don't have any bones. And they also have a large central vacuole to store a lot of extra material just in case times get tough, because they can't just pack up and leave. Animal cells, they're heterotrophic. They consume food because they can't make their own. Hence the name heterotroph or consumer. And they're mobile. Therefore, they don't need a cell wall for extra protection. They have many vacuoles, but they're small. And there's no chloroplast, since they don't make their own food. And they also contain lysosomes because they eat stuff, and it must be digested or broken down in their cell, or out of their cell in like a stomach. What do they have both in common? They both carry out cellular respiration. Animals and plants, heterotrophs and autotrophs. They both make ATP. Therefore, they both make CO2. They both contain things like ribosomes. I'm not going to go over all the organelles, but which make proteins. They contain a nucleus, which contains DNA, the instructions to make all the proteins, which control your traits. They contain vacuoles, both of them, which store material. They have the endoplasmic reticulum, which helps make fats and proteins and transport materials around the cell. And they also all have cell membranes, which is semi-permeable, lets certain things in and certain things out. And I'd be remiss if I did not focus on this one more time. Cell respiration and photosynthesis. What organisms carry these processes out? Cell respiration is carried out by both autotrophs and heterotrophs. Photosynthesis, only autotrophs. So autotrophs do both. Producers do both of these. Good luck, everybody.